Listen up, because according to the latest examiner reports, most students lose incredibly easy marks on Cambridge Biology Syllabus Topic 1.2, Prokaryotic Cell Structure. Seriously, it happens every single year. But by diving straight into these reports for this explainer, we can figure out exactly what the examiners are actually looking for, how to sidestep their hidden traps, and most importantly, how to lock in those top mark bands together. Let's dive right in.
Take a look at this. Here is a painful 15-second mistake that literally destroys your chances of hitting that top mark band. On the left, we have the flawed answer. The bacterium has a cell wall to protect it. I mean, it sounds true, right? But guess what? It earns 0 application marks. 0. Now look at the improved answer on the right. The peptidoglycan cell wall prevents osmotic lysis in the hypotonic environment described. Do you see the difference there? The absolute crucial takeaway is that using generic textbook descriptions instead of context-specific terminology is a fatal error in A-level biology.
And that leads us right to this startling number, 3. The Cambridge Principal Examiner reports highlight exactly 3 massive mistakes that completely tank student grades on this topic. It's not that candidates don't know what a bacterium looks like, it's that they walk blindly into these 3 specific traps during the exam. So let's deconstruct what they are.
Let's start with Section 1, Core Principles and Cross-Topic Connections.
Look, if you've just been rote memorizing the organelles of a typical bacterium, the reports suggest you might be setting yourself up to fail. Why? Because that approach totally ignores causal relationships. You really need to understand the theoretical architecture of the cell, not just a grocery list of its parts. Prokaryotic cell structure isn't just some diagram to label. The Cambridge Assessment Framework defines it as a biological architecture requiring connected conceptual understanding that you can transfer to completely unfamiliar contexts. They prioritize analytical depth way over superficial breadth. They don't just want to know if you can identify a plasmid, they want to know if you can explain what it does in a brand new scenario they've just invented for the paper.
We literally have a direct quote right here from the examiners that proves just how high the stakes are. Candidates referencing cross-topic connections achieve significantly higher marks. Guys, this is the hidden rule of the mark scheme. Integration with other syllabus areas isn't just a nice recommendation. It is literally how you unlock the highest grades. If you're talking about a prokaryotic cell structure without linking it to protein synthesis, genetic variation, or transport mechanisms, well, you are just leaving easy marks on the table.
So, how do we actually grab those marks? We have to look at how the evaluation works. There are 3 distinct cognitive domains targeted by the assessment objectives here. First up, knowledge and understanding, that's your baseline. Second, handling and evaluating information, that's where the context comes in. And third, experimental skills. We have to hit all 3 of these targets to truly master the topic.
Take a quick second to process the question on your screen. Soon, we're going to look at the exact examiner clue that changes your answer completely. It's basically the secret to those cross-topic connection marks we just talked about. But before we can even spot that clue, we need to make sure we're using the right mental framework.
And here is a framework to help make sense of the whole shebang. Just remember the simple acronym TUC. T stands for terminology. It must be precise and specific, like saying peptidoglycan instead of just cell wall. U stands for units. Always check and verify them, especially when calculating the size of a bacterium. And C stands for context. You absolutely must link your answer directly to the scenario given in the prompt. Using TUC will basically bulletproof your answer against the most common exam mistakes.
Moving right along to Section 2, the Standardized Technical Methodology.
This is a real game changer. This is where we break down complex problems to earn marks for the actual process, even if your final conclusion is slightly off. Welcome to the method mark strategy. The data reveals an exact 5-step sequence required for systematically processing exam questions. Step 1. Deconstruct the question and find those command words. Step 2. Extract relevant data from the stimulus material. Step 3. Select your analytical framework. Step 4. Execute your answer with incredibly clear notation. And Step 5. Present it using the correct terminology. If you follow this exact sequence, you literally force the examiner to give you method marks.
Let's see how that looks in action with the comparative analysis. This is a super common trap. When you're asked to compare a prokaryotic cell to a eukaryotic cell, top-scoring critical analysis requires identifying similarities and differences. Now, for the differences, notice the requirement here to use comparative connectives. We can't just list a bunch of facts. We have to explicitly state, for example, that prokaryotes have 70S ribosomes, whereas eukaryotes have 80S ribosomes. And then, we must draw a conclusion on the relative importance of this, like how it dictates the rate of protein synthesis.
And that approach hits the nail right on the head, as you can see in this powerful quote from the examiners. Top-scoring candidates demonstrate evaluative thinking by questioning assumptions, considering different perspectives, and justifying decisions. Evaluating evidence quality means identifying limitations and alternative interpretations. Going way beyond just regurgitating facts and actually justifying why a specific biological structure matters in that given context.
Okay, brace yourselves for Section 3, Examiner Warnings, and your interactive challenge.
Let's step into the examiner's shoes for a minute and watch a disaster unfold in real time. Here is the tragic timeline of a very common mistake. At second 1, the student reads the stimulus material. But by second 30, the panic sets in, and they rush to apply generic textbook principles instead of analyzing the actual prompt in front of them. At minute 2, they fail to check the specific units and context. And by minute 5, they've written a whole massive paragraph and lost all their application marks. They fell right into the trap.
Which brings us to an absolute golden rule derived directly from those reports. Examiner alert. Always link every point to the given scenario. It's fascinating, really. It does not matter how brilliant your knowledge of a typical bacterium is. If you don't link it to the scenario on the page, the mark scheme simply doesn't allow the examiner to give you the mark. No exceptions.
All right, let's put this to the test together. Pause this explainer for just a second and try to spot the error in this statement. The bacterium is too long and contains circular DNA, which means it will survive the antibiotic. So what's missing in this flawed calculation and analysis? Think back to that TUC framework we just talked about.
Okay, here is the red pen correction, exactly how an expert examiner would mark it. First, the missing units. Too long means, well, actually scratch that. It means absolutely nothing. It needs to be 2 micrometers. Second, it completely fails the context and terminology test. It doesn't specify the exact mechanism. We can't just say circular DNA. We need to identify it specifically as a plasmid that carries antibiotic resistance genes, and then link that directly to the specific antibiotic context provided in the question. This is exactly how you select the analytical framework and execute it with clear notation.
Which brings us to our final takeaway, the diagnostic cliffhanger. We've covered a massive amount of ground today. The core principles prioritizing analytical depth, the 5-step methodology, and of course, the TUC framework. But here is the diagnostic cliffhanger. Knowing the biology is completely useless if you don't understand the hidden language of the test itself. Think about it. If you misread one single word in the prompt, like if you describe when you were supposed to explain, your entire biologically perfect answer gets 0 marks.
Consistent practice and high-level strategy are absolutely non-negotiable if you want to succeed. So make sure you subscribe to our channel, Ace the Exams, to catch every single explainer in this series, where we decode exactly what you need to know and exactly how Cambridge wants to see it.
So what is that hidden language that can absolutely tank your perfect answer? It's the command words. But it leaves us with a critical question. How many marks have you already lost in past papers simply because you misdecoded a single verb? The next explainer on your screen right now is biology command words that decide the mark band. Click the explainer on the screen and let's decode that hidden language together.